Hey, so I have a hard mic drop at 4 p.m. So we got 1 hour. Is
1 hour?
Yeah.
1 hour.
Is that too little, Andrew? Did you have more to say?
No, I think that's probably a good amount of time. I don't want to spend too long on it. So.
Yeah. So
we probably have other stuff we can do afterwards.
So what would you like to say about Donny Budinsky and his blocking atheist policy?
How to put it into words, or friendly words.
It doesn't have to be friendly.
Friendly words, I guess. I don't know,
bad show. Is that what you would say? If you were British, would you be going, bad show, I say. Bad show.
Well, unfortunately, I'm not British, though.
If you were.
Yeah, if I was, yeah. Well, I guess just his behavior lately, well, like, I guess not really lately, like this whole past year or two, his channel has been all about the debate, not style of debate, the debate aspect of it, kind of like setting it up as if it's an us versus them mentality. And that's kind of been what he's been growing around lately. He does host a fair amount of debates anyway, so I guess that kind of makes sense. But like, I guess because before he was doing like, oh, we've demolished the evolutionists and all these 3-hour-long response quote unquote videos to other people online, but just lately he's been
bit of a wanker.
Yeah, he seems to be getting more aggressive. I don't know if it's just me, but like just more.
Maybe he's a fascist. He's just gone fascist, I think.
He's kind of
cutting off dialogue.
Yeah,
it has to be his way.
Well, I think he's probably gotten to a certain point in his channel where, and I'm just speculating here, but like he's at a certain level of power, so to speak, and he doesn't want that power to be questioned. Basically, like he has to maintain his authority on whatever he's talking about. And kind of shoot down the other side. At least that's my theory.
Well, the only reason he ever dealt with us at all is to gain subs for his channel. And now that he's got Hovind and he's got some, he's got a fair amount of steam going with 18,000 subscribers.
Yeah.
So he don't need us no more. So he just threw us by the wayside. So he's a little dodgy, or kind of an arsehole.
Should we call him
Deals, like Erica calls him?
Yeah, or we could start calling him Dodgy Donny, although it's kind of hard to say. I don't know. If anyone in chat can come up with something better, has to be
daft cow. A daft cow. That's what British would call him. Daft cow. Yeah.
James Apperson. That is SFT, standing for truth.
Wankerosity.
Yeah. Donny Deals.
Yeah.
Wait, he has 18,000 subs.
Yeah, he does. He's up to
18,000. He is growing.
Well, he's smart. He knows what he's doing. That's for goddamn sure. And he works really hard at it. So I'll give him that. But he's creating a ministry, and someday someday people are going to know who he is, but they're not going to know who Kent Hovind is. That's my prediction.
Yeah, probably. Yeah,
future. And because he's working at this ministry thing.
He's kind of the successor in some respects.
Yeah, the amazing
thing.
Yeah,
the successor and wankerosity.
They're doing it with such bad science that it's just incredible. It's atrocious. I mean, if he was doing it, based on some kind of hate speech or some other political bullshit, I could almost forgive him more than I can with what he does with science. But
I still think he has swastikas on his underwear briefs.
he does. Well, he probably did. But I want you to listen to this little part that Ron Matt says here.
Longevity, and the other one, the age of the earth. Right? How can you defend a position where the evidence is against you? I'm like, bring it on. That's exactly what we do here.
Did you hear that? How can you defend a position where the evidence is against you? Bring it on. That's what we do here. We defend positions that have no evidence, is what he said. That's what he just said.
He's just an idiot.
Yeah. Okay. It's just, I mean, that was wonderful. Now, listen again to what he says, because I think it's really precious. It's just
I like the subjects that are really hard to defend. One of those things being extreme longevity, and the other one, the age of the earth, right? How can you defend a position where the evidence is against you? I'm like, bring it on. That's exactly what we do here.
That's what we do here.
Now I wonder, by age of the earth, is he talking about an old age or a young age? Because, like you're saying, I feel like he's talking about a young age, but maybe he's
actually saying
defending it. Yeah.
Bring on your evidence, because we defend against arguments that have evidence because we have none. That's what he said out loud, but he didn't realize he said it because he is Raw Matt, the fruit bat.
A Freudian slip in some sense.
Well, no, he doesn't even need it to be Freudian. I mean, it's a matter of the fact that he's been lacking in nutrition for 20 years. He's been
a lack of self-awareness in what he said. Basically.
He's been sucking on beehives and trying to survive as a breatharian. That'll fuck you up. That screws the brain up quite a bit. I'm not sure. We got another part here. Here's the part we were talking about.
Question is, what the hell was he talking about chromosome 9? About how you can find the fusion sites calling card in chromosome 9? Because as we just saw, you can't. You might be able to find like the telomere calling cards, in the telomeres of chromosome 9. But the only place that you find the fusion site in forward and reverse, it's in human chromosome 2. Nowhere else, as we just saw. So double check me, Donny. Go see what you can find.
At the end of the day, this feels a lot like Donny just.
Okay, I almost want to play it again. So you heard her say it loud and clear. The chromosome 2 area, the so-called fusion site, the 798 base pair signature, that's the only place, she says, and she said this in her previous video where she initially made the challenge a year to a year and a half ago. And so she reiterated.
Anyway, so there are many interstitial telomeric sequences inside of chromosomes, and some of them do show signs of a fusion site or a Robertsonian fusion where there are two reverse areas. There aren't that many of those. There is one at chromosome 9, and it's located, I think, about maybe 5 or 10 million, or about 1 million or 2 nucleotides from the telomere. And there's a lot of ongoing research on this. And in fact, chromosomes have all kinds of remnants of old fusion sites.
Oh, yeah.
I've been able to find many of them in primates, and lots of evidence for not just in primates, but in all animals. Like in the insects, they found tons of stuff, and they've done a great deal of work on this to show that it actually shows you a phylogeny of how chromosome fusions and chromosome splits have happened. Now, the other way that these telomeric sequences will be inserted in a chromosome is if there's a double break strand repair, and that often inserts these sequences. But what Erica was saying is that this TTAGG is, you're going to find that everywhere because it's a simple sequence, and if you search for it, you'll find, I think, 45,000 cases of that, that particular random
I mean,
sequence.
the human genome is pretty big, so you're bound to find
Right.
that particular sequence somewhere.
And the problem is when you find one of those things, if one of them pops up randomly, and they can easily pop up randomly because it isn't that long, then if there's a chromosome break or some sort of damage to the chromosome, a telomere can very easily fold over onto that and cause basically a repetition of that unit. And of course, it will degrade after a while. But anyway, so that is not proof that chromosome 2 fusion did not happen. What is proof of chromosome 2 fusion is we have that fusion site, right? And the thing about chromosome 2 fusion is that it's a very new fusion.
Right.
These other ones are so old that you can barely find the remnants. But it just so happens in chromosome 2, it's not just the fusion site. The fusion site was
they seem to be sticking on the fusion aspect of it, and like as if we're arguing that this has been the only fusion event that's ever occurred in the history of life. Like no one's arguing that.
And what you want to look at is the fact that there is synteny, or the genes line up between chromosome 2A, 2B, and chromosome 2. And they line up in such a way that points right at that fusion site. And then when we go there, we find evidence of a fusion. I mean, that's it. It's that fucking simple, Donny. And all the shit in the world. Well, and then they go on about DDX11, which we've gone over in some detail, but we're going to go over that in just glorious detail, because I think I'm going to work on that for the next week or two.
Okay.
Because that is not a... Oh my God! What does he do here? He's got a. Let me see if I can find it. There's some other things.
But just to kind of reiterate and add on to some of the points you were just making, exactly. Like it's not just the fusion aspect of it, because like you said, you can find fusion sites across the genome. However, they may now be, but like it's. To them, it must just be a coincidence. Like the designer just happened to design it this way, that 2 chimp chromosomes of a particular length match up with 1 human chromosome that it looks like they've been fused together. Like you said, the genes are in the same particular order. The synteny matches up. The banding pattern matches up. Like you can argue, well, this particular telomeric signature, and it's not just a telomeric signature anyway. You have to look for the cryptic centromere too. Like there's clearly one on human chromosome 2. So how to get around that, I don't know. If you're just arguing that you can find these repeats across the genome, sure you can. Like there are reasons why you can do that, probably because there are other telomeres in other chromosomes too, and like you said, sequences can just happen to repeat over and over again a few times because the genome is just that freaking large. Like they're taking the non-issue, the non-issue here, and ignoring the foundational part of it. I hope I'm making sense with that.
Yeah. Hang on a second. Why did I do that? So, what was I going to say? Damn it! I was going to say. On the centromere. Okay, they found that anchor 3D, I think, gene, and they make the same claims about that. It's a highly functional gene, and it's somewhere in the middle of the. But anyway, there are reasons for that. It's the same as DDX11, and they got nothing on DDX11 at all.
Yeah. Or let me guess, it's a gene that's typically found near centromere chromosomes.
Probably. I can't remember the exact details of that.
Okay.
One,
we'll have to look at that again, I guess.
I believe it's also a pseudogene.
Yeah. I
and now, okay, evidence that DDX11L2 is the pseudogene. We'll get to that. There's a part here I wanted to play.
If you look at the sequences outside of this region, okay, the sequences are very isolated and separated by hundreds to thousands of bases. So the entire signature just completely breaks down outside of this region. But again,
I think he's just mumbling garbage there. I don't know what he's saying.
Well, I think what he's implying, and I don't think if he like was aware that he like said this, because it kind of destroys his point here that the fusion site here isn't unique in any way, because he was saying, okay, you find these telomeric repeats at the fusion site in this high density, as Erica was pointing out. Like they mentioned that and acknowledge it, but then they kind of keep it under the rug for the rest of the video.
Right.
But like he's talking about that, and then I think what he's implying, and he'd be correct on this, of course, because it's the actual fusion site, that outside of it, like the signature goes away because you're outside of the fusion site. Like there are no more, or there are fewer telomeric repeats by chance. So
yeah, and they make the point over and over again that if two telomeres fused, or two chromosomes fused by the telomeres, that there would be thousands of these things, right? No. Okay, they overlap because they're the same on each side, right? So they can overlap at any point, and they tend to overlap in such a way so that those two small arms of the chromosome are hanging out there and they get chopped off.
Yeah,
which is what we found, and then you're going to end up with a very, very small fusion site, right?
Right, because if you put a bunch of those repeats there, like they're not going to want to, for other genetic reasons, of course, but like it's an unstable configuration. Like the cell doesn't like that, so it's gonna snip out, or it's not going to be under purifying selection, obviously. So it makes sense that it, like, this is their other major point in this video, that, oh well, it's too highly degraded and it's not pristine enough compared to these other sites in the genome, if those are indeed fusion sites. But
the site he shows in chromosome 9 is also highly degraded.
Yeah. Well, like he makes the point that it's like right next to each other, touching, but like, yeah, it's still highly degraded. Like it's again not in the, it's not a bunch of these sequences right next to each other in this high density configuration, like Erica was saying, and as he's showing in this diagram here. And I feel like he must have lined that up wrong, because I'm pretty sure it's higher, a higher number of those sequences in that section there than what he's showing. So I could be wrong. Yeah. But like if you just change out like one of the letters here and there, then it does become the signature sequence. So like there obviously have been point mutations and deletions at the fusion site itself, because again, it's no longer under purifying selection. It's like the cell doesn't care what happens to it anymore, because it's not a telomere anymore. It's not protecting the chromosome.
Yeah, I've got a picture of it here. Let's see, the actual fusion site. And I'm showing more TTAGGs than he's showing.
Right, yeah, exactly.
I'm not sure. Oh, shit, I took the wrong picture. Let me just try to get a picture up here and take a look at it. Okay, there's the fusion, and I have to go all the way out there to the coding gene. And we'll wait for that to cook. Let me see here. Where are you? Come on. You can do it, Mac. Just put it up there. Don't. Oh my God. Why do they do that? I don't know why they do that. Let's see here. PTSC. Okay, here's the fusion site that I have. Now, is that he's showing 1, 2, 3, 4, 5, 6, 7, 8. And I'm showing 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. What the? Why do I have so many more than him?
Yeah, I was like, did he line it up?
I don't know. Because that shows.
Yeah, and as Nestle points out here, at some point you do supplement the TTA motifs. Like, why do you see that if it's not a fusion site? Like, he hasn't explained that.
Well, he thinks it's highly functional, like God planned it.
Yeah,
and the other thing is, God was really tired because he made all the animals, and he spent a long time on the chimpanzee because he had to make them just like this human thing, right?
Right.
And then God forgot to hit return between chromosome 2A and 2B. So he forgot to hit the return key. So they ended up fused in humans because God was really tired And it was like nearing the 7th day, right, when he needed to take a day's rest because, he got tired, right? So that's why he forgot to hit the carriage return. That's why there's a chromosome 2 fusion.
Yeah.
I don't know why they just don't say, well, because it's a miracle.
Can you, is this like a pre-highlight sequence, or can you highlight the CCCTAA inverse complements too?
No, this is a picture that I took. I have, like, I did this stuff like 2 years ago, 2 or 3 years ago, 3 years ago.
Okay.
The very first time I talked to Donny, I showed him this picture. So I'd have to go and find this stuff. But I'm doing all of this research over again. So
okay.
It's just that it takes a lot of time, and I don't get $120,000 or $140,000 a year like Jeffrey Tompkins does. So I don't get shit. I get shit.
But I mean, doesn't at some point he says, Donny, I mean, that he wasn't borrowing from Tompkins' work, but his diagram looks like the exact same thing as what Tompkins showed in one of his other papers. So.
Yeah, I think he got this at the creationist site. He got that chromosome 9 stuff from the creationist site too.
Yeah, probably. Yeah.
He claims he did his own blast. Well, he said we. He said we, and he was referring to him and Tompkins, right?
Right.
Like, I think it was just
I thought it was him and Chris Roop. He said.
Maybe, I don't know. Not sure.
Yeah. I guess it doesn't really matter at this point.
Right. Anyway, so there's something else here. Let's see if there's a few
healthy telomeres. Actually, Matt, if you wanted to cover this argument briefly, brother.
Healthy telomeres.
Healthy telomeres are thousands of bases long. And so if you had 2 healthy telomeres slammed together to form a fusion, we should find thousands and thousands of bases. But
No, for lots of reasons.
Yeah,
Basically, they overlap. They can slide anywhere. They can slide like 6 at a time to any length, basically, and get truncated. And that isn't exactly how they work. They form this structure. It's almost like a crystalline structure with loops, and that's why the Robertsonian fusion happens the way it happens.
It's almost
And
like
it
he
leaves a
doesn't
very small star.
It's almost like he doesn't do enough studying of the topic, and then he makes wacky whack-a-mole conclusions based on incomplete and incorrect information.
Oh yeah, he does that all the time. Yeah, that's a common thing with him.
Yeah, they go on about that a little while, and then there's something else here. Okay,
there's actually something he said about nested hierarchies that we can get to later, but I think it demonstrates what you just said, Scott, because I don't think he knows what he's talking about again. But sorry.
Now I want to find out where Matt talks about testicles, because that was pretty fun, and maybe this is it. Let's see.
And in the genome, we've discovered that there's significant activity and function associated with them.
No,
then why isn't the fusion site just a higher dense region of functional activity in the genome? I mean, we understand, Matt, that the fusion site region comprises what? It comprises multiple start points for transcription, where RNA polymerase II binds. Okay, it's an amazing source of genomic activity. We've demonstrated that over and over again. And the fusion site itself, which isn't a fusion site as we're proving here, is essentially this functional component inside an essential gene, the DDX11L2 gene that's completely on fire.
Okay. Oh my God. So he's claiming that this fusion, that TTAG, that telomeric sequence, he's claiming that is a functional part of the gene, the functional part of the highly functional DDX11 gene. But it's in the intron, number one. There's nothing functional about it. The fact that it's in an intron only occurs on some transcriptions because there's one little tiny exon called exon 1 that's way out on the 5' end, and that sometimes gets translated with the entire DDX11 gene, but not always.
Right.
And
it's usually the transcripts are outside the fusion site.
Yeah.
Right?
The other thing is that I think there are somewhere between 14 and I think 32 variations, human variations on this gene, where the exons are all over the place, indicating that it is a pseudogene because it's all over the place.
Right.
And if it's let's see, if some of those mutations are involved in a particular kind of genetic defect. I forget what it's called. Starts with a W. I forget what it is here. I don't have a note on it. Anyway.
Before we move on, can we just highlight Nestlig's latest comment here? Because I really like the analogy he gives here. I think that's a very good analogy for it. Yeah, Donny looks at the wake of a car crash, complains that some of the material of the original cars are not there anymore, so there was no car crash.
Right.
Yeah, exactly. Same point.
Yeah, that's pretty good.
Yeah.
That is really good. Oh, let's see here. So I wonder if Raw Matt starts up here. Raw Matt says a really wonderful, funny thing here. I don't know. We'll just let it run.
Ooh, nice.
And highly expressed. Matt, what do you say to the evolutionists that argue, oh, well, the DDX11L2, it's a pseudogene. The activity there is just spurious. It's not really functional. You guys are just making that up. It's a good response.
I would have to say, okay, well, first of all, where is it expressed and how is it expressed? You'll be able to find multiple regions in the genome where it's completely expressed.
Here we go.
In high amounts, actually. It's just expressed very different because we also find that same gene in other things, and it's expressed very differently. So I thought I would have had it in my notes over here, where it is actually expressed and how much. Very strange, I don't.
I just want to point out that what they have here is from that the Harmonisome study, where they did all sorts of interaction studies of all kinds of proteins. And it's just a massive study. And they found 1,996 what they call functional associations. And they're not functional associations. That's badly named. They are the fact that these proteins stick to another protein or another something somewhere. They found some sort of stickiness among a protein. Now, I could make a random protein and it would come up with 500 or maybe 1,000 functional associations in any cell in the body, basically. So, I mean, the interactivity of proteins is immensely networked and complicated, and it happens all over the place. And this is just not an argument. Now, if you search for DDX11L2 in Google Scholar, you'll find a very small amount of pages, 6 pages, and all 6 pages only refer to it in reference to somebody that found out that it had a stickiness or an association at some point in their study, but none of the papers talk about DDX11L2 exclusively. None of the papers have found a known function for this pseudogene. And this pseudogene is only a fragment of an actual DDX gene. And it doesn't have, like DDX11 has it's not a dead box. I call it a DEA box. It's a DEHA box. And if I'm not mistaken, there's not even a dead box domain inside of that DDX11. The only thing they found is that it has some similarity to the tail end of a functional gene, DDX11. And that's why they think it's a pseudogene, because it doesn't have enough domains to do anything. And I've shown the picture, and I've gone over that before, and it's about 1/5 or 1/6 the size of that full gene. It's just not a fucking gene.
Yeah. Well, like the whole thing about the DDX11L2, like it actually makes a stronger case for it being a fusion site, because, like, again, this family of genes are usually found
on telomeres.
Near the telomeres.
Yeah.
So so if it's found near the fusion site, that kind of makes it even a stronger case that these telomeres did fuse together at some point.
I just wanted to look at Raw Matt's face on this.
Now, Nestlig has put a link in the side chat that you might want to just go ahead and throw in the description.
Yeah, he's saying standing quote minded. Surprise, surprise.
He quote mined?
I know, shocker.
I thought honest people of integrity didn't do that.
Okay, the Yuxin fan paper is the famous key paper in all of these things, and he's been talking about it for 4 or 5 years. So that's the one.
Oh, okay.
It's kind of the main genomic structure and evolution of the ancestral chromosome fusion site. There's been a lot of work done since, and And as of 2023, there are no papers that indicate there is any function to the DDX11L2 gene. None.
Yeah.
We have nothing. So they keep saying highly functional. But listen to what Raw Matt says here. Oh my God. All right, we have sound effects now.
Oh, sorry.
I'm usually pretty good with my notes. Apparently, I haven't done this for a while, so
Yeah, you're doing good, brother. I pulled up so people can look at this paper. We looked at this ourselves. We looked at it with C. Brown as well, who helped us, because again, the critics. Dr. Jeffrey Tompkins, he's done a fantastic job. He's done a sophisticated analysis of the DDX11L2.
Yeah, no.
But the evolutionists hate creationists, right? The second they hear,
yeah, because he gets this like evidence from the Bible first, and that's why they don't really like you. You know, you can't do that. You're either going to go with science or you're going to go with the Bible. You can't do both. You can't publish fucking paper.
I take issue with him saying like, oh, we hate creation. Like, I don't hate creationists. No. Like what I do hate is the way they act, the way they portray science, the way the dishonesty that they have sometimes. Like their behavior. That's what I'm frustrated with. Like, I don't hate them as a person. Like if they could change, then yeah, then they'd be a better person overall. So.
Yep.
I just take issue with that statement, but whatever. Do we want to maybe see if Neslick wants to join? He seems to have a thing or two to say here beyond what we're saying.
We don't have time for other people to join.
Oh, that's right. You got
drop dead in 25. I have to get out of here in 25 minutes. There's absolutely no way out of it. So
sorry, Neslick.
Sidetrack. Okay, so right here is where he got that from. Okay, this is the harmonizome.
Okay.
People can read that or not. DDX11 has 1996 functional associations, blah, blah, blah. And they show, basically that from all these different studies, they found that this protein sticks to something. That's all they found. Now, if you look at DDX11, which is not the pseudogene, okay, that one has, let's see here, implicate. Okay, DDX11 has 4,085 functional associations with biological entities. Okay, that is the full gene. That's the full protein that actually does something. It has something to do with, I think, transcription or RNA or DNA repair, I think.
No, is this the gene found in chimps?
No, it's in humans. DDX11 is in humans. I think it's on chromosome 12. The actual.
Oh, it's a different gene.
By the telomere. Okay. Yeah, it's a different gene, and there's I think there's a family of DDX11 genes.
Yeah.
It's actually it's not a dead box, but it's a dia box gene.
Yeah.
Sometimes it's dead, but anyway, okay. So there's that. But there's something wonderful that Matt says that I want to get to.
Do you want to take this comment down real quick?
Do what?
Can you take the highlighted comment down real quick?
Yeah.
There we go. Okay. So it's not blocking anything.
Oh, let's see. Where did I bookmark this thing? Oh, my bookmark program. Blue. Or maybe it's here.
Did this research. It's like now everything.
Yeah, maybe it's here. Let me try this. I got it somewhere. We'll just find the part where he talks about testicles.
See at the bottom where those associations are and how much in the heart, the gallbladder, the body fat, the spleen, the testes, where it's actually a lot of it is concentrated. And it's just amazing how much function that they have in these regions, which means it's vitally important. If you find a lot of function in the testes, that means that it's obviously inherited. It's passed on. It's de novo, right? That's how.
Did you hear that? Did you hear what he just said? If you find a functional gene in the testes, it's obviously inherited. Passed on. And then he says it's de novo. Like,
does he know what de novo means?
This guy will just say anything. I mean, he'll just it's like his brain just starts leaking words.
Like this is stuff that you can, like, if you had basic high school biology, like, and like that's kind of what I'm using here, but like that's, come on.
Okay.
If you knew this stuff, you'd know that what he's saying is just
Okay, I just want everybody to hear this again, and then we'll go a little bit further and see what else he
Right.
At the bottom where those associations are and how much in the heart, the gallbladder, the body fat, the spleen, the testes, where it's actually a lot of it is concentrated. And it's just amazing how much function that they have in these regions, which means it's vitally important. If you find a lot of function in the testes, that means that it's obviously inherited. It's passed on. It's de novo, right? That's how important it is. It's not something even like an orphan gene, something that it arises later when it's epigenetically needed. If you did a knockout,
is he saying de novo or bonobo? I don't know what he's saying.
De novo. I don't know. But okay, and then he goes on and starts babbling about orphogenes, epigenetically when it's needed.
Like, did he say it? Like, does he think it's created like through epigen? Like, does he even know what epigenetics means?
Okay, now one thing I want to point out that everybody needs to listen to is that the testes. Now, if you look at all genes, or you look at even pseudogenes or just little random chunks of DNA. If you look at a graph of what tissues they're expressed in, the testes is always the very highest. For some reason, in male testes, everything expresses. There's like it's like no holds barred. It's no wonder that we get testicular cancer because there's a whole lot of expression in the testes, and that doesn't say anything about how functional it is.
Right.
That just tells you where it is expressed. Now, being expressed is not the same as being functional. Sticking to something is not the same as being functional. Functional is when you find an actual mechanism of function that does something that's really important. And then we know that we have a functional gene. And until we find
none of that means it's going to be inherited, despite what he said.
Right. Just because it's in your testicles, it's going to be inherited. He's talking about, he's confusing it. He thinks everything in the testicles is automatically in the gametes.
Right.
So he thinks it like leaks into the gametes, right? And it's like, this is the funniest shit I've heard. And I'm only like I mean, I only went about 5 minutes further. And this is just wonderful. I mean, this is raw mat gold.
That was pretty good. Yeah. Thanks for clipping that.
I don't know. Anyway. I wonder if I have any.
Yes, we have we satisfied your need right now, nerdy rodent?
Hold on a minute.
So I want to thank. I guess we'll speeds away. Thank you, everyone in chat, for joining us today. Hope you're doing okay. Hope this has been an entertaining
news as we are. I mean, these guys, they're refuting fucking evolutionary biology. They're refuting millions of researchers and millions of papers, and they say things like it's in the testicles, so of course it's inherited.
Right.
Like,
they remind me of those on Star Trek: The Next Generation. Those aliens that came to the Enterprise, flew into the vicinity of the Enterprise, wanting spare parts or whatever, and they just kept going, "We're smart," and they would do stupid things,
Yeah.
Smart.
Yeah.
And
and actually, he does look like Spock, kind of. He looks like, doesn't he?
It looks like Spock after like a transporter accident.
He's got pointed ears. I mean, an pointed head. And
well, like more relevant to hear, like what it reminds me of is like, you remember that one time where Dr. Dan went on with Kent Hovind and asked him about the 5 mechanisms of evolution, and he couldn't do it. Just to kind of show that he doesn't know what he's talking about. Like, that's kind of the exact same thing that's going on here.
I taught high school biology for 14 years in the basement of a church where they invited me.
Right.
Yeah. I taught high school biology for 8 years to my sons, so I'm a biology teacher. Anyway, I don't know. I'm not sure. Let's see. It goes on, but we are in a hurry. So 12 different, not spurious. I don't know what the hell. Oh, yeah, I'm not sure.
Even imagine that you, Matt, as a human, are related to a banana plant. That's science, Matt. Matt, you don't believe you're related to strawberries and banana plants?
Well, I'm glad the law doesn't, because I would be a cannibal. I'd be eating.
I just wanted to point out that they keep doing this related, because when humans, like if I ask you, Andrew, are you related to Scott? You're going to say no.
Right.
Right. So they keep, like, they love to push that on their people. They've got a bunch of brainwashed people that are lapping this stuff up, and they always like to say dumb things like, are you related to a banana plant? Well, I'm not related to you and Scott.
Right.
I'm
Yeah,
sorry.
this is comic relief in creation world.
Yeah.
So you got a bunch of Christian YouTube viewers are going right now because of what they're saying.
It more speaks to the fact of how intelligent they think their audience is, and all that.
Do you guys want to go on without me? We could let Nessie in, and you can go on as long.
How do you have to head out?
I have to head out at 4.
Okay. Would Scott be able to end the stream?
Yeah.
Okay.
Well, I'll probably need to go at 4 too, because of those particular commitments I have this evening.
Oh, right.
Okay,
You've
fair
got
enough.
a babysitting commitment. Yeah, I'm sorry, Andrew. It's just that this just came up.
But it's not even 4 yet. We still have 15 minutes of raucous comedy to bestow upon our listeners.
I'm going to send Nestle an invite in case he has anything to add here.
Yeah.
Because I got out everything I needed to get out. I just wanted to show the comic relief.
Yeah, if you want to come in, Nestle, and give your thoughts for the next 10, 15 minutes, that'd be great.
I think at some point I'm going to debate Standing for Truth on Chromosome 2 on his channel.
That'd be cool.
Because I did it like years ago when I was just getting into all this and I wasn't prepared.
Yeah.
And I still kicked his ass, but he insists I didn't, because he mentioned satellite DNA and I didn't know what the fuck it was. So anyway.
Well, like, doesn't he still use that argument anymore, despite the fact that it's used to identify specific people for the very reason that that's highly variable?
Right. Yeah. Among
that doesn't
within
even
the
make
species.
any sense.
Yeah. And within primates, the satellite DNA regions are just all over the place.
Right.
Chimpanzees it's like really degenerate in chimpanzees and all over. And so that if you compare us to chimpanzees, you'll end up with differences. But he makes some kind of case that the satellite DNA should be in a certain spot, and that too doesn't make any sense. It's like they have these little nuggets, and I just cannot. Yeah, okay. So Nestle linked the Mayan Lab Harmonizome gene project.
Oh, okay.
I should put that in the description. I'm not sure if I'm going to have time today, but I'll get it by tomorrow. Okay, let's look at it. Let's look at insulin. Okay, he's got one for insulin. How many? Open link in new tab.
And of course, later in the video, he brings, kind of somewhat unrelated to the chromosome 2 fusion. He brings up the nested hierarchies and cars again, and then he says, and I have a feeling that maybe he got this wording from me, or maybe some other film about this, because I don't think he was saying it before, but he said he's in this video that cars and automobiles, they naturally fall into nested hierarchies. Now correct me if I'm wrong, but he wasn't using that terminology before, right? So
yeah.
Has he kind of picked up that again without understanding what it means in this context? Phylogenetics and whatnot. I don't know, just a hunch.
This is the insulin one. There's 7,433 functional associations. Well, I'm sorry, but insulin does not have 7,433 different ways of acting. It has an effect on chemistry, because all chemicals do, and proteins are chemicals, and they stick to every goddamn thing.
Right.
I mean, just the fact that they stick to something, which is what these data sets are from, does not mean, and most of this is from ENCODE, right? ENCODE went through and did an exhaustive stickiness study. The stickiness is also what gives us the massive amount of variation from which natural selection can operate on to create new mechanisms and create new information. So this is why we evolve, because of that unbelievable cross action between all sorts of different proteins.
Yeah.
And if they get in different regions and they happen to have a functional benefit, they may end up in a population, and they may end up going through a body. Oh, by the way, did anybody see Dr. Joel Duff's thing on Scott? What was it on? It was on a gene duplication in mating species of moths, I think, and they
no recollection of that.
Oh, you sent the link to me, for Christ's sake.
I know that because
it was 2 days
You
ago.
would remember it. I figured you would remember.
Yeah, it was 2 days ago, so I can understand why you don't remember.
It just seems like something that you would really, really be interested in. So that's why I sent it to you, man.
Yeah. Anyway.
Okay, Nestlig is clarifying here. You can see where the gene is most expressed. Insulin is very specifically expressed, while DDX11L2 is all over the place.
Okay, I've been instructed to tell you to click next to expression in tissues and cell lines.
Oh. Do what?
Click next to expressions in tissues and cell lines.
Yeah, I'm looking for it. Oh. Here we go. Okay. That one gives you. Okay. So this is insulin. Okay. And we've got, it's pretty specific. Okay. Let's go back and look at DDX11.
Yeah, let's compare them.
For that one. Yeah. It's all over the place, basically.
Yeah.
Because it's a small gene, and it's going to express everywhere. Interesting. Thank you, Nestlig.
Yeah, thanks, Nestlig. He's providing some good links today.
He is a source of good information.
Yeah.
Yeah, and I got to get these into the description before we close out.
Hey, Scott, has Swifter been in chat before? Or?
Maybe a few times.
Yeah, I've seen him around a few times. I don't know how often he frequents this channel, but thanks for coming on, Swifter.
And there's CRISPR, CRISPR-Cas9.
Hey,
good to see you,
CRISPR.
sir.
Haven't seen CRISPR in a while.
Oh, let's see.
You've been summoned by the magic words. Which magic words, I wonder?
Yeah.
I'm not sure what the magic. Oh, chromosome 2, I think is the magic word for CRISPR.
Oh.
Or gene.
Darn, I wish we could have CRISPR on. That'd be great.
If you say gene 3 times, I wish we had time today.
Yeah.
Okay, so let's see here. I want to back up.
The P.T. Barnum saying really goes here. Speed, always leave them wanting more.
Yeah.
Right.
There you go. Oh, well, at some point we got to get serious about this chromosome.
Oh, is Grignard reagent? Hey, good to see you again.
Oh, same one.
Yeah, you remember it? Yeah, cool.
Changed his name.
Yes, CRISPR was summoned by chromosome 2.
Okay. Anyway, they don't have. I don't know. I haven't watched the rest of the video. I'll have to check the whole thing out. But I don't think they have much more. I'm putting links in for this harmonizome database, which I think CRISPR hates. Kind of.
Do you think CRISPR would be able to come in for like a few minutes in case he has something quick to say?
I don't know if I have CRISPR's email address, but we can put the link out.
Yeah.
But I got to leave I'm leaving in 10 minutes, and I think Scott's leaving in 10 minutes. And that means we're going to end the stream, no matter how rude I got to get, even if I just got to end it right in the middle of somebody's sentence.
Okay.
So just as long as everybody knows that.
Yeah.
Harm database doesn't show function.
Yeah, but it uses the word just like encode does, and they shouldn't do that.
Okay, so it's misusing a term.
Yeah, functional is just a bad word to use unless you can prove some sort of mechanism for function. Healing flow.
And here's the stream that I originally got it all. Well, sort of. Like, because Erica responded to parts of this one, and then now Standing is responding to her. How far back it's.
No, this is. I think this is. Oh, there's Steve Baumann. He has a thing out today on Is Genesis History.
Yeah.
I should link. I'll link that. Hang on a second. I'm busy. Steve Baumann.
Busy as a bee.
I've been using my descriptions to find things because I am so goddamn good at doing a description that if I go back into a certain period of time and I'm wondering where the hell was that video, there's that violin thing with Hillary Hahn, Scott. And this is Dr. Joel Duff on the, this is the Dr. Joel Duff video I mentioned. Pheromone receptors and moths, and it's really cool because like a population of moths that's all breeding together, right? If a few of them get a mutation in these pheromone receptors, they're more likely to breed with each other, or basically that creates a speciation event among moths, and he explains all that in this video, and it's done with a gene duplication on the one he's talking about, and it's pretty cool. So it's worth watching for anybody that wants to dig in deep, because he goes pretty deep on this paper. So Dr. Joel Duff is always amazing.
Yep.
And who else we got here? Okay, this is, there's Nerdy Rodent. I'm going to watch a little bit of that. Oh, no.
Are those rocks?
Oh, my gosh.
Who would believe you could freak out in Minnesota?
Wow.
That's
did that?
That's a line from Frank Zappa, Mothers of Invention.
Yeah.
I don't know if that was Call Any Vegetable, or I forget what song that was. But Call Any Vegetable was an amazing Frank Zappa song. The vegetable will respond to you.
Okay, Nessling has a little more to share. Something else, while insulin is highly expressed exclusively in pancreatic cells, one could respond that DDXT blah, blah, blah is something that's more general functional in the cell lines. In all cell lines.
Okay, now he's following up. However, I looked up a housekeeping gene involved in actin cytoskeleton. It is needed in all cells. Expression levels of this gene, again, unlike DDX11L2, it's highly expressed in all cells. So yeah. Can't make the case one way or another.
The way to make the case as to whether or not it's expressed is to knock it out, I guess, but they don't want to do that with humans, so I don't know, but I would volunteer for that knockout experiment because I don't think I need that piece of shit. I just don't want it. And I'm old, it won't matter if I start, stuttering or whatever. Or
yeah, here, CRISPR's saying what you were saying earlier, Speed.
What's that?
About the testes and spurious expression.
They have a lot of expression of almost everything you look at. You'll have a high spike for the testes. If you look up a gene on NCBI and you scroll down a little bit, it usually shows you a tissue expression graph, and you'll find the testes sticking out there just as, like in a lot of ways, our testes stick out into places where they shouldn't be. And that's one of them. So, who in the hell thought of that design, putting them on the outside? Like, imagine naked men running through the bushes with those things hanging between your legs. It's not a good fucking idea.
And that's for clothes.
I've done this, and I tell you, it's not a good idea.
Hey, this is kind of a kind of related to that, but kind of an off-topic thing. Did you know there's a clade of mammals called Scrotifera because they have external scrotums?
Oh no!
Or at least I don't know if it's still accepted as an actual clade at this point, but I know it was at one point.
We have to look into Scrotifera.
Look it up real quick. Check if I'm right or wrong or not.
That can become like an expression. Like for something's really great, you can call it scrotiferic.
Okay, it means beasts with scrotum. is a clade of placental mammals that groups together order Chiroptera and grand order Frungulata and something else. But
okay, that sounds about right. Yeah,
You know, I've seen a few like in the rest stops on the highways across the land. I've seen a few beasts with scrotums.
yeah.
yes, I've been to some sauna parties.
So we're kind of humans. We humans are unique in that regard, in that we're not part of Scrotivora, but we also have external scrotums.
And they hang way too far. And it's just pretty sad. Pretty sad. I don't know what the hell they're for. I have a whole theory on that, Scott, about women and where women came from, but we'll get into that in another stream another day, because we got to end this one.
Yep.
So I put stuff in the description. Okay.
Okay. Thanks everyone for joining us today. Hope you found this interesting and entertaining.
Yep.
As always, from Standing for Truth.
There's nothing like a science power hour on Answers in Atheism.
Yep. Okay, boys and girls, buffaloes and bunnies, and everything else. Goodbye, cruel world.
Bye.